if I'm going to ask somebody to do something, I should be prepared to do it, right? So these days, they don't ask me to do much. They try and keep me out of the labs, but certainly if anybody walked in here this minute and said, hey, we need something done, I will roll up my sleeves and go get it done. It's important as well, so I'll divert a little bit. People are important, right? Every single person who works for this company, I don't look at them as just that person. They have a family who's dependent on them, right? So we've got to be able to work together to make it successful for not just the number that are employees of the company. It goes further than that. So, yep, I will roll up my sleeves any day.
Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello, and welcome to BioTalk Unzipped, where our mission is to unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin. I'm a life science BD professional in bioanalysis from Kansas City. And along with me from North Carolina is my friend and co-host, Dr. Chad Briscoe. Chad is a bioanalytical scientific leader, and we publish BioTalk Unzipped at the beginning of each month and bring experts from different corners of the pharmaceutical, biotech, and medical device worlds to share their innovations, insights, career journeys, and personal stories to the show. Please subscribe and tell us what you'd like to hear about in the future. It may show up in a future episode. And thanks for listening. Today, we have the privilege of hosting Dr. Alfred Botchway. He's a distinguished figure in the field of biomedical science with over 25 years of experience spanning academia, pharmaceuticals, and contract research organizations. Dr. Botchway is the CEO of Attentive Science, a non-clinical CRO specializing in vivo services to support pharmaceutical, animal health, agrochemical, and food product development. Dr. Botchway received a PhD in cardiovascular physiology, and he is the past president of the International Safety Pharmacology Society and the past board chair of BioKansas. Welcome to the show, Dr. Botchway. How are you?
Great. Thanks for having me. I'm glad to be here with you guys.
Well, we appreciate you joining us. And I want to thank Chad for inviting you on. And this is our first time getting a chance to talk. Wanted to kind of start, from the beginning. Tell us a little bit about what inspired your initial interests in biomedical science and your educational journey. How did all that kind of shape your career path to where you are today?
Great question. My interest really started, obviously, when I was a kid. Had an uncle who was a medical doctor, and I tried to aspire to be him, right? So right from a very early age, I knew I was going to be an MD. But as life goes on, and for most of us, as you learn more, you think, well, you don't necessarily have to be an MD to help science, right? So one of the things I struggled with was really being too empathetic. I got to a point where I realized there was no way anybody was going to show up at my practice and tell me they had some disease and I wouldn't go home and think about it and maybe even get that disease, right? So the way for me to help science was to go into research and ended up doing multiple degrees. Maybe it took me too long to get to where I needed to, but yep, got there eventually.
Well, all things work out in the end, don't they?
Okay.
Very good. And you've had some really cool experiences in business as well as the science. What were some of the challenges that you faced, while founding Xenometrics, and kind of how did you navigate that transition during its acquisition?
Hey, to talk a little bit more about the business side, the founding of Xenometrics really was a necessity, which, long story short, migrated from Denmark to the US, got here 9 months later. The company I came to work for decided they were closing their doors, right? So I had an option to go back to Europe or try and do something. So I had the opportunity to start Xenometrics, and really it was a little bit of luck because if you think of it, the company had just closed down. There were people resources that were looking for jobs, and I somehow was bold enough to say, I'm going to start a company, got together with some of the old folks, and off we went. And it's been over 20 years.
I think so. So, Alf, having known you for a number of years now, I guess going back probably about 15 years, I know there's a lot more to it than that, right? I recall certainly, hearing how you guys sort of carved out a corner in one office in the building, and you started with what you know, and it's a pretty inspiring journey, actually. I think, to where, like you said, it was kind of, partly out of necessity. You had other options, but you said, hey, let's take a shot at this, and and see if we can grind it out, and that's something that I think you're under selling kind of the achievement that you guys had.
I try to keep it short there, but yeah, I can expand a
Yeah,
little bit more on that.
yeah, please.
So literally the day I made the final decision to start the company, I had movers come into KC to relocate me to another state because I had another job in, gosh, I think it was like North Carolina, maybe.
Yeah.
And I literally had to call the guy who had just offered me the job to say, you know what? I need to take a chance on something. And if it doesn't work, I'm going to come crawling for you to get me back the position that you just offered me. But if I do not do this, I'll never know. So roll the dice, turned down, called the moving company to tell them not to show up, and went back to the facility that had just been closed and had a word with the management to say, I believe we can do something to fill the gap that you currently have. At the time, I can now say, because they no longer exist at the site, this site was owned by Bayer Crop Science. This was the Center of Excellence for Toxicology, North America, for Bayer Crop Science. They kind of looked at me funny going, you really think you can do this? I said, yes, I can. So they gave me a really tiny corner office in a 60,000 square foot facility. And as we progressed, we kept asking for additional rooms to lease from them. And finally, they realized we needed the building more than they did. So we ended up buying the assets after 3 years in being in business to own all the assets and the book of business. And the rest was history. So to your point, Chad, it wasn't literally overnight. There was a little bit of sleepless nights, and we finally got there.
Well, then I think what's impressive again is I saw you do it kind of all over again in a little bit different way when you started Attentive Science, and what's maybe was more impressive to me even the second time around is I remember actually visiting when you guys were setting up Attentive, and you and your son Michael were in there painting yourselves, right? You're like, we're going to start this business. So we have a guy who starts a business, highly successful CEO, sells the business, could certainly have opportunities all over the place, and you're starting up this new business, and you say, you know what, I still roll my sleeves up and come in, I paint it myself, nothing is too, nothing is sort of beneath me, and again, I think that's something that I've been impressed in the time I've known you. And now it seems that Attentive is doing quite well also, right? So on at least your second go round with really success there.
Yep. We're certainly doing good. And that principle, I think, was instilled in me years back to say, if I'm going to ask somebody to do something, I should be prepared to do it, right? So these days, they don't ask me to do much. They try and keep me out of the labs, but certainly if anybody walked in here this minute and said, hey, we need something done, I will roll up my sleeves and go get it done. There comes a time as well in this kind of career path where you crave the science, right? It's not just sitting at your desk looking at spreadsheets, and it's important as well. So I'll divert a little bit. People are important, right? Every single person who works for this company, I don't look at them as just that person. They have a family who's dependent on them, right? So we've got to be able to work together to make it successful for not just the number that are employees of the company. It goes further than that. So, yep, I will roll up my sleeves any day.
Yeah.
That's great. That's great. Oh, I'm curious, what do you love about what you do now with Attentive? And how do you feel that's making an impact?
One of the reasons I think I've stayed in this space is the variation, right? We get to see all sorts of modalities, all sorts of companies starting. What I really take pride in is working with a company that had an idea, and then later on you hear they've been bought for 2.x billion. Of course, I don't get any of that money, but my gratitude really comes to the point where, oh my gosh, yeah, I remember when they had their first compound and they didn't even know what to do. And 10 years later, here we are. So that's really what keeps me going.
That's excellent. In, I believe it was 2023 did a presentation, and you talked about AI's role in non-clinical safety assessments. How do you feel about where it is today, and where do you foresee AI transforming this field in the next 5 to 10 years?
We've still got a fair bit to go. The AI, one of my other passions is messing around with computers and computer programs, right? So yeah, I like playing with the AI side of things. Gosh, I've got so many opinions on this. So where do I start? Digital twins, right? I like to look at that in a little bit more of a simplistic way. So forever in true research GLP environments, you've got to have a control group, right? Those control test systems, I believe, have a huge potential in really amplifying this whole AI space. There are some industries as well, which prior to actually conducting work for them, they would ask for historical database. We as an industry have a huge amount of historical database given to the right folks. I believe will be decades ahead of where we are right now. But the complication is everybody believes they have their IP and don't really want to share the data, but the control data is control data. It's what dosing with DMSO. That's not really the IP. Trying to convince our industry that there is a huge amount of value in that historical database is what I believe will propel us into the next level in this AI field. And really, when we talk about AI, some people think it's going to replace their jobs. I don't believe so. It's literally being able to sit back and look at the data and really understand it. That's what's going to make the difference. The AI aspect allows us to look at it quicker and to be able to get to conclusions. So, yeah, that's it's another favorite topic of mine going, really, what's AI? It's enhancing what we do. And how do we really get our hands around it to get drugs to the market quicker? And in my case, make sure that the toxic side effects are not showing up again and again because we have the data.
Yeah, so that was one of the AI, I guess, is a new tool in the 3 Rs, right? We talk about the 3 Rs a lot in the safety testing space, and replace, reduce, refine, I believe is what the 3 Rs are. And how do you see AI playing a role in that? You alluded to it a little bit, but specifically in that context.
Yeah, certainly. So refinement would really be the most important thing for us in the non-clinical toxicology space, right? How do we compile all the data? And I know this sounds a little bit crazy, but because of confidentiality, new clients who come to me saying they want to do this, I can't necessarily share everything that I know from that kind of refinement space, but we try and advise them to, even reduce the number of test systems that they're using. We all went to some sort of college, university, we're trained to increase the number of subjects in our studies, to increase
the power.
Right.
Yeah.
Yep.
Yeah. Statistical power. Yeah.
Do we still need to be doing that? Right. We've done this for decades. I think really this comes to changing the mindset, right? We've all got to know that we've got enough data power now to be able to refine our studies and still be confident in the data that we get. So yeah, refinement, reduction, the 3 Rs are really important.
Yeah, I think it's interesting. We talked with Tom Helikar, I don't know if you're familiar with him, at the University of Nebraska, about his digital twin program that he's developing. And, we have talked a lot about different AI modalities really across this spectrum. But I think as you mentioned, it will enable us to really get to the trends in the data analysis a lot faster than we've ever been able to do.
The answer quicker. And so I've got to go back to something on the same topic. So the failures in the non-clinical space is valuable to our industry. But again, we don't talk about them, right? We only talk about, company X is now in phase 2, phase 3. How many compounds did it take for them to get there? That data set is what I personally would love to get my hands on, right? To really understand why did they fail. And through some AI modeling, be able to get to those answers quicker.
And in I've never thought about this before, but in the clinical space, you have to report failed studies. Do you have to do the same in the non-clinical?
No,
Okay.
it just gets shelved.
Okay. Yeah, we just had some interactions about a failed study that that's what happens sometimes. But the FDA came back and wanted some more information about it, so it's a real thing, and it is on a level it's being looked at for clinical trials within the FDA, but I'm not sure. I don't think anyone is aggregating that data to look across at this point.
So the FDA is obviously aggregating data in the non-clinical space as SINT, right? Standard for electronic non-clinical data. But again, I don't believe they have all the data that includes the failed ones. And if you are an FDA person, I'll tell you, can you please give us access to the data that you have, right? I mean, they're using it for purpose to be able to approve the drugs that go into clinical trials. But from our side of the table, we're never going to see that information, right? So, and the FDA database that's open enough and obviously confidential information scrubbed would be really helpful to the industry.
Yeah, that's it's a challenge on the FDA side. I think probably if you were to talk to the individual scientists, they would be in favor, but then they're part of a massive bureaucracy that's getting no less complex for sure.
I believe there are multiple consortiums that are trying to do this as well, aggregating data on the non-clinical side. The few that I've tried, I've not been impressed, probably because there wasn't a real standardization of how the data was input, right? But you dig, and you go, nope, the study said this was the result, but it really wasn't, right? So there's still a lot of work for the human to do.
So companies like Attentive work according to the GLPs, the 21 CFR Part 58, and again, I sit alongside that. I certainly know the bioanalytical pieces of it that are critical, but I was wondering as I was prepping to talk to you a little bit today, where does that, where do those regulations stand as far as at one point there was kind of a modernization, I think, because they're originally in 1977 and then started trying to modernize the GLPs. Has that taken place? Is it underway? What needs to be done there?
When it comes to GLPs, there shouldn't be so much modernization or change, in my opinion. Gosh, all of a sudden I'm thinking of the Theranos stories and people.
Yeah.
I mean, it comes down to documentation being attributable, legible. Anybody should be able to walk into any organization and really be able to tear that study apart and understand exactly what went on, right? So that principle should never be touched, right? Or else we're all in trouble. Every question you ask, my mind just goes all over the place. If I'm to kind of talk about some of the APAC countries where we don't believe the research is that solid, I've had personal experience going to visit some companies in China, and gosh, there's no single reagent on the desk, but when you open the drawer, they all expired, and you go, what are you really using? Oh, no, we're not using those. Really? Come on. So good laboratory practice should not be altered in terms of documentation. Training is the other thing, right? We do have folks who come into the industry who know nothing about GLP, but the basic notion of what they're doing has always and needs to be GLP, right? You're in a lab, you've got to document things and make sure anybody who comes in after you can attribute it to something.
Yeah, that's yeah, I like that philosophy, because I think even with part 11 in 1997 being released, it was kind of, people really have went, oh my goodness, what is this? But really all it said was, it said a few more words, but it said, hey, the same rules apply when it's a computer versus when it's paper and pen and paper, right? It's but just make that transition, so those key principles don't really change. So yeah.
Very good. Well, I think, Chad, it's time for new news.
New news.
Oh, new news?
Well done.
Very good.
All right,
Published
let's hear it, Greg.
yesterday in Nature is an article entitled "How the Brain Suppresses Fear. Mouse Study Offers Path to Anxiety Treatments," written by Felicity Nelson. And this study, which is originally published in the journal Science, explores how mice learn to suppress instinctive fear responses, revealing two key brain regions involved in the process. So researchers found that while the posterolateral higher visual areas help mice to learn to override fear, the ventrolateral geniculate nucleus, or VLGN, stores the memory of the learned suppression. And so they were able to kind of switch on or off the using light as the posterior high visual areas, that memory of the fear. But what was interesting is that this unexpected result was that the VLGN was not previously considered a region capable of plasticity or memory, but they were able to, the ones that had learned more courage, that this object that was coming in was not a predator, was able to remember that even after they switched that part off. So it suggests the potential implications for anxiety and PTSD treatments in humans, particularly through things possibly like deep brain stimulation or targeted modulation of endocannabinoid activity. Curious how you feel about this. How could findings from this study maybe shape drug discovery efforts for anxiety and PTSD? And what are your feelings around endocannabinoid uses as a treatment?
Oh, wow. This could be another 2-hour discussion. It's the psychedelics, right?
Oh, yeah.
Yep.
Psilocybin, yeah.
Right,
Yeah.
yep. So yeah, there is a lot going on the non-clinical side of research. Of course, fear and excitement. That's a whole field that there's lots of research that's been published and done. I'm going to take you a little bit on a tangent here in terms of science versus what really happens. Personally, I live on those two every single day, right? I mean, literally, you either respond to a certain stimulus in one way or the other, right? You can decide it's something good when you get that stimulus and deal with it, or think this is the worst thing that ever happened to you, and then all your systems are going to shut down, right? So, yep, back to your question, how do I think that's going to affect our space and research moving forward? I really think there's more to come, especially when you think about the psychedelics, right? We've done several studies recently where these test systems, you can see them being on a trip, right? And it's kind of cool to think, oh my gosh, I wonder what they're thinking about and what states they're in, right? So the CNS space is really interesting.
Yeah, that's it is really interesting. And I think it also, when you think about the fear and the fear and those responses and how they relate to anxiety, which in some ways, I guess it is the same and it isn't the same, and it's all part of some different emotions. But also it kind of goes back to why there's so much attention paid to the environment where the testing is done, that there's proper care, that all are as comfortable as possible, right? It's similar again with the clinical trials. You want everybody in a great state of mind when they're in these studies, and that they're not in that fear response, because even if your drug is not dealing with fear response, your drug may be affected because of an altered state that you're in. And as I'm speaking, just like more, you said this could be a 2-hour conversation, but or more. But there's whole conferences around this topic, right? But I'm thinking, do we look enough at how anxiety and other conditions affect just how you metabolize drugs, right? Does one metabolize a drug differently because they're in a very anxious state, or because they're in a different state of mind, or not feeling well, and how does that affect it? And I'm sure it's different with different organ systems as well.
Absolutely. All our organ systems are connected, right? Gosh,
Yeah.
if it's whatever state you're in, anxious, excited, your heart is going to do something, right? Coming back to the heart of the matter here being, your heart is racing and other systems are completely affected. So yes.
Pretty fantastic.
And I heard, and this goes back many years, back when I was in college, I was auditing a course in neuroscience, and the professor, I really remember this because it really landed for me, why stress is so exhausting, right? And so he had discussed a study that demonstrated potentially that the mitochondria under stressful situations were working up to 10 times as hard. And that makes sense, because you think you could be sitting all day, right? But if you're in a state of cortical stimulation and you're stressed out, that you could be exhausted even though you didn't do anything physical, right? Now, unfortunately, I haven't been able to track down that article. I've been searching. I even got a hold of the professor, and he couldn't remember because that was, he just mentioned it during class. But have you ever heard anything like that? And does that seem right to you?
I probably would have heard it, but I was smiling because I'm thinking, gosh, that's my daily life, right? How do I deal with the stress? And if I allow every single stimulus that comes to me to be stressful, I will be stressed. I mean, there's no two ways about it. So one of the things I say to people is, you got to feed your mind with the right things, right? If you feed your mind with stressful things, you are going to be stressed. Just like the body needs food, the mind needs some nutrition. And depending on what you give it, your end result is different.
Garbage in, garbage out.
Yeah. Now, you mentioned something that I'd maybe like to spend a couple of minutes to discuss. So you talked about academia. One of my challenges these days to myself and everybody I get to meet is, how do we find the next toxicologist, the next pharmacokineticist, the next safety pharmacology person, right? I don't believe we're intertwined enough with academia to let them know that this industry exists. I ended up in the industry again by chance, right? Presenting at some conference, some pharma company guy walks up to me going, yeah, I think what you're doing is interesting. At the time, it was when a lot of drugs were hitting the head channel, causing torsade de pointes. I was in the cardiovascular field. They said, come help us make sure our drugs are not causing this problem. So off I went, right? But if that particular day had not happened, I'd be, I don't know where I would be right now, right? So for us to have kind of made that shift to industry, I keep asking, how do we reach back into academia to educate our future scientists and make sure that they know this is a field that's important and exists and they can contribute to it?
It's a really important point. And I've tried to impress that upon my 15-year-old who has a little bit of interest. We sent him to like a pharmacy camp at KU for a couple of days to be exposed to the field and what the program would look like. And he was kind of sort of interested in it. And he's tangentially maybe interested in pharmaceutical science. But I agree. I think it's almost the best kept secret, and it shouldn't be. That's out there in the industry and in the science. I think you need to start with the high schools and I think the hard part is trying to speak to them at sort of where they're at and explain to them, how cool it is, these innovations that you can work on, and these treatments that dramatically can help people's lives, maybe even save people's lives. And it's a very wonderful, stable career as well.
Absolutely.
It's interesting you say that about high school, Gregory, because for me, I always thought I was always fascinated with science, but science was generally my least favorite classes until college, even. I mean, some aspects of my high school science I found interesting, but the learning and I probably none of my teachers from those days are listening to the podcast, but apologies if they are, but they don't generally do a great job making science interesting at the in the high school and middle school, right? I think there's so much more that can be done to engage kids in the wonders of it, and probably worrying less. This is probably across a lot of fields, but probably worrying less that you learn everything, but learn how to learn how to love science, learn how to be really fascinated by just a few areas instead of trying to know something about everything, because that's not going to stick. You're not remembering chapter 10 of 8th grade biology class or whatever it
Now,
was.
Chad, you're touching on so many things, and you've got my mind kind of racing again, going, yep.
Yeah.
If I asked most people with a PhD, including myself, right, do you use exactly what you learned for your PhD every single day? The answer is no, right?
Nope.
You will use pieces of it as you go along, but not just that, right? And being able to broaden your mind and think logically about different things based on that training. One of my professors said to me, and I will never forget this, he said, getting a PhD is really showing to yourself that you can start something and finish it. It's not really coming down to the exact topic that you did. Gosh, I was stuck in a Faraday cage, doing all sorts of electrophysiology forever. I haven't seen a Faraday cage in a while, right? It's now all, what do you call it, ion channel instruments that you buy, you plug in, and off he goes. But yep, the data that comes from the other side, you've got to be able to interpret.
Yeah, without a doubt. I mean, there's the grind, of the PhD, there's the thinking process, and I think that finishing is so important, because that I think that is maybe the hardest part for almost everyone in their journey. Is just getting through the end. It was certainly was for me. I was lucky to have a lot of people that were kind of kicking my butt, saying you just got to finish this, right? And lucky to have that, because if I didn't have it, I don't know that I would have gotten through it.
Wouldn't it be cool if we change our mindset to be more of a vocational training, right, for our future scientists? Because the more time they spend in the lab, even pipetting, right? That's what's really going to make a difference. And going through the academic sludge is, it's tough, right? We've got to do it. I'm not saying let's get rid of academia, but it's a double-edged sword. You've got professors who don't really want to let go of their students because they write grant applications, and that's what funds the students. They get to buy more instruments, and they're literally holding on to people when industry needs them, right? So,
for sure.
Yep. And I think it might help motivate students to decide to advance beyond a bachelor's as well, if they understand what it is they can do and what their career opportunities are and what impact they can have. And if they don't know what they don't know, that may not drive them to go to the next level, unless they happen to, kind of fall into a lab situation and they get exposed to it, and then they get excited and, do it from there.
Yeah, it's, I don't want to sound hypocritically, but I did it. I did a bachelor's, master's, and then the PhD. And then when I did a DSP, it's like, come on, how many degrees do you need? Right? And at some point, you got to cut it.
Yeah, for sure. No doubt. So that kind of goes to one of the other topics I wanted to talk to you about a little bit, Alf, is your community involvement. And that's a couple of different types of communities, right? I, as you know, I first knew of you from my brother. He actually, had mentioned a few things about you. I think it was when I was moving to Kansas City. He said, I know a guy in Kansas City that you need to meet, and and as soon as I met you, I knew you were the guy. But so there's that, that kind of community, and your involvement in the Safety Farm Society, and then also, your involvement in the local Kansas City area life sciences initiatives, BioKansas, and I think there's been some other things. Can you talk about why, you know, why you're involved in those, why you think they're really important? Why are those the areas that you've chosen to invest your time over the years?
The very basic answer is being able to give back. The two local organizations that you mentioned, well, maybe three of them. So BioKansas, and there's also an organization called Pipeline Entrepreneurs. Really, when I was trying to find my feet, invested a lot in me, right? Moving from Europe to KC, where do you find scientists? Yeah, you try and go to the BioKansas get-togethers, the events, and get to know people. So from a very early stage, I believe they invested time in me, trying to understand what my aims and objectives were. And I ended up serving on the boards of those organizations for probably too long, until they had to change the bylaws and say, no, after a certain number of years, we've got to kick you off, right? You've been here too long, right? Yeah. So BioKansas, for example, was a great one. I was on the board for several years and ended up being the chair of the board. My CEO at the time left, so I had to become the interim CEO, which kind of coincided with my non-compete 1 year period. So I had to find something to do. I wasn't going to sit at home.
Kind of convenient.
Yep, it did.
In a way.
And managed to find the next CEO, and the organization continues to grow. Like every organization, you've got to adapt and change. So there's been changes for the best. But yeah, I'd gladly go back and support them any day. To my point about who's the next scientist, where they're coming from, that when it comes to BioKansas in particular, I believe that's where the next industrial scientist goes to, right, to try and find out what's going on. So, yep, I'd gladly be there to talk to them and tell them my story and how they can get there.
I was just going to ask also, how, you're you're an interesting guy and you've done so much. You've got this diverse background. We haven't even talked about your connection back to Ghana. And if you don't mind, talking about maybe how that, I think your mother still lives in Ghana, if I recall correctly, and how that connection, right? I mean, so we have Ghana, we have the UK, we have, Denmark, the US, and Kansas City, right? But going way back there, how does that inform who you are today?
Where do I start? Obviously, my parents instilled in me some very good qualities to say, try and be the best at whatever you do, right? So in any situation, in any country I find myself, I'm always trying to not just elevate the group of people that I'm with, but I want to learn something that I can take further on. Talking about my mom, yep, she's still alive. She's 92 years old. She's the sharpest thing you can ever come across. Do not have a conversation with her this week and think she's going to forget next month, because she will remind you, right? She somehow may have also contributed to my kind of entrepreneurial journey. My mom started a nursery in her house, looking after 500 kids between the age of 1 and 5. At some point she had up to 400 of them. They all call her grandma. Those kids have grown to be doctors and God knows what right now. They have gone out. They come back and visit her, and are forever grateful for the very first few years of molding their life. And I was one of them. I was the last of 6. So by the time I came around, my elder brothers and sisters had gone through all the necessary training that they could hand over to me. And I had a great upbringing. I go back at least once every year to see the old lady. And interestingly enough, over the last 5 years, I've been going back with a group of at least 12 or 15 people, most of them who have never been on that continent. They trust me enough to say, hey, we're going to come along with you. We go, we have a good time, we get to explore the country and also look at other business opportunities. So I've got some of those colleagues that have gone with me that literally are now naturalized. They have businesses over there and would have never left the US. they've probably gone back more times than I have been, because there is some value out there. And the value out there, if I'm to maybe expand on that, is youth.
Sure.
There's a lot of youth on that continent as a general, and the youth is eager to learn, right?
Yeah. Unlike in the US.
Bringing it back to our science that we talked about, the people who do best in our field are those who are eager to learn, right? You get technicians, some of them want to progress, they learn quicker because they want to learn, right? So the key important attributes is who's willing to step up and learn something that they don't know about, right? And that makes a huge difference.
Oh, it's really cool.
Yeah, it's awesome.
And I'm curious, you've given us a lot of clues to your leadership style. You're hands-on, you're willing to get your hands dirty, you care about people, you understand family is important. How much of an influence was, your upbringing and your mom into your current leadership style for your company?
Oh, okay. Which one do I want to touch on? Because there's so much going on in the world these days. Like, okay, where do you go? Where do you not go? Just talking about doing your best in every situation, right? I'm gonna say then, and I'm gonna loop back into the biggest kind of discussion these days being DEI, right? It took me a while. I think it was about 3 years ago when I applied for, gosh, what's that program called? It's like a minority business entity certification. And going into this whole process, I thought they were looking at just me as the minority. But as you go through the process and you get to answer the questions that they're asking about the whole organization. So then you have to describe how many women of different ethnicities. And all of a sudden I'm looking at my organization going, oh my gosh, we are the most diverse organization you can ever come across, right? So going back to your question of how did the upbringing, I've always been in a place where you could consider me being different, and it never made any difference to me because I've been brought up to acknowledge that I was different and I was there to be different for a reason, right? So that's I think what's kind of all tied up into where I am today and how I look at people in the organization, how I look at my colleagues in the industry, and let's achieve together.
Yeah, that's great. I appreciate you sharing that. And what are your strategic goals for Attentive looking into, the future and where you want to go from here, and how you want to continue to address some of the emerging challenges in the biopharmaceutical industry?
The 2 hours, if I could describe it, but I'm going to try and keep it short. So most recently, Attentive Science has gone global, Australia and Abu Dhabi. And the question always comes up, why those two regions? So I'll start with Abu Dhabi. There isn't a lot of non-clinical research in that kind of North America, Middle East region. A lot of their focus has always been in the oil industry. They've got a lot of money, but then they need to diversify the industry. Not only do they need to diversify the industry, but there is a little bit of a genetic concentration, which they are doing something about. I mean, I believe they're going to have all their folks sequenced in the not so long future. So specific therapies for that region becomes important. How do we help that region to be successful and treat the diseases that are applicable to that region, right? There's got to be some scientists who are willing to take the step and get over there and work with the folks that are there to do it. And Attentive Science is going to be one of those companies. We've broken ground. We're going to have a building completed probably in about 14 months. We have an initial small lab that's going to do some in vitro tox to start with before we get into the in vivo space. Okay, so that's the kind of Abu Dhabi region. When it comes to Australia, so Australia is known for clinical trials. They're ahead of us for a while when it comes to clinical trials. But if you look on the non-clinical side, there's very little, right? So they're having to export some of their requirements. And from my opinion, there are challenges when it comes to non-clinical studies. If you talk about certain test systems where you've got to ship samples back to Nebraska for analysis, the CITES permits, there's all sorts of things and regulations that we've got to abide by, right? So being in that region is important for attentive science, and especially when it comes to the test systems that we will be addressing. So we're going global to help other regions, to encourage growth in those regions. And at the end of the day, I believe if we do our job right and we continue to focus on helping each other and helping science, we'll all benefit.
So are you encouraging some of your own staff to see if they're willing to go there, to train, to live,
if you know,
like permanently or temporarily?
To go to any of those regions, let me know.
Wow.
We've got 2 that have relocated already to Abu Dhabi, and we've got 1 that's about to relocate to Australia. So yeah, and you know what? Maybe that again, talking about how I used to say to my kids, if anybody asked you where you're from, just tell them you're from the world, because you cannot explain your parents' origins and from your mom's side or your dad's side. It's just too confusing, right? So being able to step out and go to another region to live there, you do learn a lot. These regions have their advantages, and there's a lot more to share.
Australia doesn't surprise me as much because I do know about their work in clinical, and I haven't heard much about non-clinical or discovery. But Abu Dhabi, how did that come about? I'm really curious.
Oh, okay. So one of the folks who used to work for me here in the US is actually from that region. So through various conversations and what's going on there and keeping our eye closely on what they're trying to do is how we ended up there. And of course, we had to pick a place. It was going to be Dubai or Abu Dhabi. Abu Dhabi had a little bit more of the academic infrastructure, which we would need to tap into. They were a little bit more forthcoming in what they wanted to do for the region, and it turned out to be a place for us.
Makes sense. Yeah, I don't know much about Abu Dhabi. Been to Dubai once, and Dubai, just from my observation, seems to be more about the oil industry, about commerce, real estate,
lots of
tourism.
shiny buildings, right?
Yep, big shiny buildings, moving those cranes around.
Yeah. But I think that region, I mean, obviously there's a lot of money there for investment, and I think they're continuing to look to ways to diversify. So it actually seems like a pretty smart move. So, I mean, I'm sure, Alf, you'd like to think so. And I wish you the best of luck. It'll be interesting to watch where that goes.
Thank you. Yeah. We've got work to do, but we'll get there.
Yeah. Very good. Very good.
Does the government at all, get involved in terms of, hey, we'd love to see more of this diversification and support what you're doing?
Absolutely. Yep. We're planning on working closely with the government, and when you say the government, the Department of Health in the UAE is very keen on making sure they move this forward.
Well, Alf, it's our tradition here at BioTalk where we will take a question from a past guest, not knowing who's coming on next, to ask that person. So Nathan Tusher was on earlier, and he was discussing clinical pharmacology, and he'd like to hear about innovations in sample collection, shipping, and handling for clinical trials. And he was curious about our advances in blood spot analysis, on-site analysis, et cetera, making an impact on clinical trials, not knowing that we're going to talk to a non-clinical person. So can you take that question and kind of bend it into your world? Is in your space using less blood samples or fewer test subjects to reach the same endpoints? Do you see that happening? What advances can you share in that context?
Yep, that certainly goes back to the 3 R's that Chad asked about earlier. Being able to use or sample less of a blood volume from our test systems really goes a long way. Talking about dry blood spot and all the other technologies that are out there. I'm not the expert in bioanalysis. I may know just enough to be dangerous, but that I leave it as that. So we really depend on our colleagues like Chad and his company to be able to say, how much do you need? How much of a plasma sample do you need? And once we get that to them, they provide the data of the content of the test substance in that plasma. It comes back to us and we incorporate it into our reports. You've kind of opened a different conversation for me, being sometimes the test substance that we receive doesn't really go into solution, right? So there are all these formulation craziness that has to go on before we can really administer it to the test system. And then on the bioanalytical side, we want to see data so we can correlate it to any of the effects that we see, right? We cannot always assume we're given a 5 milligram per kilogram dose when actually by the time we administer it is 3.5, right? So getting the data back from our colleagues on the bioanalytical side is important. So the technology that they use to get there is, I believe, becoming more sensitive and helping the industry as a whole.
Outstanding. Well, from the bottom of our hearts, thank you so much for your time today. It was a tremendous honor to have you on the show, to meet you. Since we're local, I've got a Guinness with your name on it. We'll have to get together in the near future and chat some more, continue this conversation, and go from there.
Great. Thanks for having me.